Hello dear friends, welcome in Pharmaceutical Chemistry-II laboratory. Myself, Mrs. Urmila Ravindra Gaudi, MPharm in Quality Assurance. Today we are going to deal with Systemic Qualitative Analysis. Experiment number 5. Title of experiment: to identify the given organic drug or compound D2 by systemic qualitative analysis, and the compound is salicylic acid. Learning objective: intellectual skill. In that we study to determine concept of the experiment, to understand the test procedure, to analyze and interpret the observation, to plan the experiment. And then motor skill: ability to write systemic qualitative report, ability to handle equipment, take and record observation, ability to refer standard literature and Indian pharmacopoeia, ability to do work to the plan of the experiment, and ability of group working. Then let's start with concept of today's practical. First, preliminary test. These are the primary tests carried out to get some idea or clue about the compound. No definite conclusion can be drawn from this test. Proposition second, physical constant. In that we have to perform melting point or boiling point determination. It is characteristic distinguish physical identity of the organic compound. Proposition 3, that is elemental analysis. It is find out to all the elements present in the organic compound by some color and precipitation reaction. Proposition 4, group analysis. It is to find out different functional group present in organic compound. Functional group is the group of element present in compound. Proposition 5, literature for the references. It is a reference table in which compound are classified according to element, groups and physical constant. Proposition 6, confirmative test. These are specific color reaction or preparation of simple derivatives and determining its melting and boiling point, which identification of the drugs. Now, what is the general concept for the structure? First we have to start with preliminary test, then physical constant, means determination of melting point or determination of boiling point, group analysis, element detection, result with reference to literature, then confirmative test. After confirmation we got the identification of the given compound. Now let's start with apparatus used for actual procedure. Glassware, test tube, beaker, measuring cylinder, graduated pipette, bulb, evaporating dish, water bath, Thales tube and thermometer. And the chemicals required are all general and table reagents. Now let's start with actual procedure in Pharmaceutical Chemistry-II laboratory. Now let's start with our actual procedure for identification of organic compound by systemic qualitative analysis. For that we have to first go through preliminary test. This is our compound, which is white crystalline powder, almost odorless. Of given sample in water. For that, take sample and add water and check the solubility, whether it is soluble in water or not. Shake vigorously. So the given compound is soluble in water. Then check solubility in acid. That is NaOH. To normal NaOH. Take sample, add to normal NaOH. Shake it thoroughly. compound soluble in 2 normal NaOH. Then check solubility in reagent, that is cold NaOH. For that we have to take 0.2 to 0.3 grams of sample, then add 2 ml of water and 2 ml of 10% sodium hydroxide solution. Add 2 ml of 10% sodium hydroxide solution. Then check it. The compound is soluble in reagent solution, that is 10% sodium hydroxide solution. Effervescence of CO2 may get evolved. That means the compound is strongly acidic compound. Then next test, action of hot concentrated H2SO4. For that we have to take approximately 0.1 gram of sample and add 1 ml of concentrated H2SO4. In that, blackening of solid compound evolved with carbon dioxide solution. That means the compound is strongly acidic. Next, action of sodium carbonate on given sample. Again take sample approximately 0.1 to 0.2 gram. Then add 10 ml of 10% sodium carbonate, which is freshly prepared, and add it into the given sample. Observation, the effervescence of CO2 evolved. That means the compound is strongly acidic. Then next test, action of KMnO4 solution. This test is used for finding the compound whether saturated or unsaturated. Take 0.1 to 0.2 gram of sample. Then add 10% sodium carbonate solution, 10 ml. Shake it properly and add KMnO4 solution drop by drop. The purple color get changes and it becomes brown. That means decolorization of purple color. That means the compound is unsaturated. It is action with APCl3. Again take approximately 0.1 to 0.2 gram of sample. Add water. And add APCl solution dropwise. Observation, no formation of violet color. That means the compound is strongly acidic. Now next test, heating on oxidized copper gauze. For that take wire gauze and add sample on it and heat it. Observation is sooty flame. That means the compound is aromatic. Now next, heating on clean glass rod. It shows Sudhi flame, that means the compound is aromatic in nature. Now next test is heating with soda lime. For that we have to take 0.5 gram of sample, then add it 2 gram of finely powdered soda lime and 1 gram of coarse soda lime. After this observation is it evolves odor of benzene, that means the compound is aromatic in nature. After heating, the odor of benzene is evolved, that means the compound is highly aromatic in nature. Determination of physical constant. Our compound is solid, that means the compound we have to take physical constant melting point. The procedure for determination of melting point we already discussed in experiment number 2. I had similarly taken the melting point for given sample, and the given sample melting point is 158 degrees Celsius. Now let's start with element detection. Procedure for this: dry small piece of take a small piece of sodium metal into the boiler tube, sodium fusion tube, heat it, and then add powder, and again heat and add it into porcelain containing 10 to 15 ml of water. We already done that procedure and kept it on heating for 10 to 15 minutes so that the sodium fusion extract get ready for the detection of element like nitrogen, sulfur and halogen. After boiling the mixture for 10 minutes, we already filter that solution, and this filtrate is known as sodium fusion extract filter, and this filter is used, sodium fusion extract is used for detection of nitrogen, sulfur and halogen. Let's start with actual procedure for determining the element. Let's start with test for nitrogen. Take 3 to 4 ml of sodium fusion extract. Then add it ferrous sulfate, approximately 0.2 gram of ferrous sulfate crystals. And 1 ml of sodium hydroxide. %% sodium hydroxide have to add into the solution, 0.1. And then boil this solution gently. After boiling, we have to add it into dilute sulfuric acid solution. After boiling, we have to add dilute sulfuric acid solution into it. Okay. No persist blue color is developed. That means the solution does not contain, the sample does not contain nitrogen. Test for sulfur. So sodium nitroxide test. Take 2 ml of sodium fusion extract, then add 1 to 2 drops of freshly peppered sodium nitroxide. Shake vigorously and observe the result. It does not show purple or deep red color. That means sulfur is absent in the given compound. Now procedure for determination of taste of halogen. We have to check halogen whether present in the compound or not. For that, take 2 ml of sample, then add HNO3, that is nitric acid, dilute nitric acid. For the solution becomes acidic. After addition of nitric acid, solution become acidic. Then add silver nitrate solution. Only 1 to 2 drops we have to add into this solution. Let's move towards determination of functional group. Now our compound containing carbon, hydrogen and oxygen element, therefore go through carboxylic acid test. First, sodium bicarbonate test. For that we have to take 0.2 gram of sample and add saturated sodium bicarbonate 2 to 3 ml solutions of saturated sodium bicarbonate. Add it into test tube containing our compound. Shake it properly. There is an effervescence of CO2. Shake it vigorously. The compound is dissolving given sodium bicarbonate solution, saturated solution. That means the compound is strongly acidic and the compound may be carboxylic acid. Now second test, ester test. We have to take sample 0.2 gram, then add 2 ml of ethanol. Shake it properly and add 2 drops of concentrated H2SO4 solution. And then solution is heat for 2 minutes and observe the boiling water bath. On boiling water bath, the fruity smell of ester is produced. That last procedure we have to conclude or confirmatory test we have to perform. For that we have to take procedure for neutral test solution. For this we have to take sample approximately 0.5 gram. Then add 1 drop of phenolphthalein indicator. And add 0.2 ml of ammonia solution. After adding ammonia solution, we have to boil this solution so that the excess of ammonia get evolved. We have to cool this solution and then add few drops of ferric chloride solution. The solution is cool now. Then add ferric chloride solution. The purple color is developed. That means the given organic compound is salicylic acid. The salicylic acid is confirmed by this confirmatory test. After performing the practical, let's move towards result. On the basis of the test performed above, with the reference of handbook, the given organic compound is aromatic in nature, unsaturated, acidic, and carboxylic acid group may be present in the compound. The melting point of the given organic compound was found to be 158 degrees Celsius. The given organic compound found to contain carbon, hydrogen, and oxygen element. The given organic compound found to contain carboxylic acid as a functional group. Conclusion: the given organic compound was having 158 degrees Celsius melting point, carbon, hydrogen, oxygen element, and carboxylic acid as a functional group. Therefore, with reference to literature, the organic compound may be salicylic acid. This is the structure of salicylic acid, and it is used as an analgesic and antipyretic drug. Thank you, dear student, for watching and attending the practical session. Thank you.